Yeah, welcome to the Cambridge Cheminformatics Network meeting. I'm Nola Boyle, and thanks once again to our host CCDC for having us here, and also to Andreas for putting us together and organizing this. So without further ado, let's begin. You're still welcome. So the fire notice is very important. So if there is a fire, follow the green exit signs, and people from the CCDC in particular will know where to go. And hopefully you've all signed in. And then when you leave, remember to sign out. So there's assembly points outside.
Got a message at the top, by the way.
There's a message at the top. Maybe I'll get rid of that. Good point. And now I've gone back to slides. Oh, no, there we are. So what if you've come here by accident, just to be sure that you're in the right place, this is a meeting which is bringing together everyone interested in cheminformatics and beyond, scientifically and personally. We've in-person components, and it's also online. I'm going to have 3 talks, followed by socializing, which is either in person in the Panton Arms down the road, or there's an online Zoom thing, socializing event organized by Andreas. This is the 3rd meeting this year. There'll be another one on 12th November. And there's the websites. It is being recorded, but per agreed with speakers, the talks will be made available online. So the questions should be typed into the chat window. And at the end of the talks, we will go through these as well as the local questions. So as said, if you're online, if you stay online after the meeting, then there's a virtual breakout room where you can meet with other attendees. And of course, Andreas is always looking for more presenters or new presenters, so get in touch with him if you're interested. So this is the program for today. So we're going to have 3 speakers. Mike Ree is going to be up first from Idea Pharma, talking about innovation in pharma. Sonha is here in the room from Johannes Gutenberg University, talking about cross-modality learning of cell painting and transcriptomics data for small molecule activity prediction. And finally, Andrew Skalkin from Datagrok will be talking about Datagrok Swiss Army knife for cheminformatics. I think that is the last slide. So, Mike, if you want to start sharing your slides.
Thank you very much. I'll see if this works. Does it say that I'm screen sharing?
Yes.
There we go. Okay, so come through okay?
Yes, I believe so.
Fantastic. Yeah, keep me honest and then tell me if I'm doing anything wrong. So yeah, and you know, I recognize this as a cheminformatics meeting. I'm going to talk almost at the very highest level about what innovation means, how we measure it, and how to get.
Sorry, there's one point. There's a little box on the window, perhaps. Is that on your side? It says continue to share this PowerPoint window.
Continue. Oh, okay. Continue to share. Has that gone away?
Not yet, but maybe if you click again.
It says,
okay. Or can you move it, or move it to one side, perhaps?
No, it's not letting me do anything with it. I'll try.
Yeah, if you just stop and maybe do it again, it'll go away.
Okay. Okay.
Okay, that's it. Thank you.
Okay, thank you. So yeah, so this is I'm going to say quite a high level presentation. I'm going to try and make it in 20-25 minutes. And the concept of innovation is something that I've been keen to talk a lot about, because I think our industry doesn't tend to do it very well. I will share these slides so you can have them offline, so don't worry about jotting anything down as we go through. Things like cartoons and stuff in here are all shareable as long as there's some attribution. But I'm really coming from the perspective of Idea Pharma, where we advise companies who are in early phase on how to get to market and how to get there successfully. So it's really keen that we establish the kind of terms of reference. And one of the questions I always asked in our industry, you know, I've been in it for like 30 odd years now, is what do we mean by innovation? And it's a term that we hear every day. It's a term that we all throw around. And actually our industry tends to discuss innovation in terms of, you know, what's interesting and what's new versus, you know, what have they created meaningful value from. But let's get very specific. Invention is not the same as innovation. And all the way through the kind of the references on innovation, this term is used very specifically and very carefully to mean the kind of creation of value from invention. And there's some great quotes here from Matt Ridley's book about how innovation works, about, for example, the very famous Edison invention of the light bulb. And as he points out, you know, that was the easy part, was inventing it. Lots of people had done, mostly independent of each other. 21 different people had invented the light bulb, but really he was the one that brought the manufacturer into the kind of realm of possibility. You know, things like the mercury vacuum pump and a bunch of other things. So you know, which is more important? Is it coming up with the idea, or is it really putting it into the hands of people and really making some, you know, creating some value from it? But that's where the kind of classical definitions of invention and innovation separate. So at IDEA, we decided it was kind of important for our industry to understand this stuff, because we invest a lot of money in exactly that. So we define innovation very specifically as the return on invention. So the creation of meaningful value from invention. And obviously we don't have to wait until launch for that, as we see in a lot of deals in biotech now. The creation of value from invention is shown there. Invention, we all recognize that. We all know that's what we do most days, you know, bringing ideas or technologies together in a novel way to create something that didn't exist before. And clearly there's a whole realm of IP that sits next to that. But I want to focus really on the innovation side of the spectrum. So, you know, we decided at IDEA about 15 years ago that this was a kind of interesting question, is one company more innovative than another in our industry? Because we tend to treat our industry as a set of averages. So we have, you know, average cost of R&D, average returns, you know, what is a blockbuster and so forth. But the closer we started looking, really, there was no average pharma company. So we said, well, let's dig into some of the kind of metrics that we might use if we want to rate the pharma companies for their effectiveness at doing the job that we all come to work to do. So we started looking at, well, look, innovation clearly involves launch, so efficiency and effectiveness in reaching the market with an asset. But then we're looking for some kind of asymmetry in the performance. So do they create greater value than a similar competitor? And we're all watching, you know, the GLP-1 war play out at the moment. We've seen PD-L1, PD-1 wars. You know, often classes have victors and people that don't do so well. So we all sort of understand the innovation side, you know, fairly well. And then clearly on the invention side, what we want to see and encourage our companies doing meaningful development, so addressing key unmet needs. You know, we're not so excited about the Me Too's and so forth. Clearly have their space and clearly have value in our industry, but that's not what we're looking at in the invention side of it. And clearly novelties or first in classes or breakthroughs. But I mentioned this kind of question that we asked ourselves, and the key question was, if you gave the same molecule to two different companies in early phase, so phase one, would they be equally successful? And everyone around us knew that the answer was no, but no one really knew why. And we thought that we knew why, and we started to get to, well, really, what are the enemies of innovation? And I won't really go through this cartoon because it was illustrative of one of the problems that we see in our industry, which is the TPP tends to constrain thinking, but also gives different people in the organization control of the molecule at different stages. And really, the idea of developing a molecule towards a phase 1, phase 2, phase 3 transition, or even to an approval, tends to neglect or forget the kind of the requirement that that molecule has to generate some revenue when it actually gets there. So the idea of designing with the end in mind, or designing with the patient in mind, is often left until much later in phase. So if I go forwards, really the idea of doing the right things and doing things right, which is this kind of classic Venn that we're all shown, which tends to lead to efficiency. It's not good enough for our industry because one of the things that we want to have on the way to becoming effective, or really driving some focus and agility into our teams, is really learning what the right things are, and really obviously the asymmetry. The faster that you can get to that place of understanding what the right things are, makes both of those other circles much easier to achieve. Clearly, pharma has this huge range of opportunity to do this learning, where the right things are. So not only, what the molecule might do in terms of its range of effect, but what might the market want, and clearly those two things are not the same. So what a molecule does, what a market wants, are not the same. What else might we put in there? So some digital or some biomarkers, or other solutions, formulation solutions and so forth. What else is possible to deliver this molecule to meet this unmet need in the marketplace? Even given an A and a B, so getting this molecule from A to B, we might choose different paths to do that same journey. And some of those will be more expensive, some of them will be faster, some of them will be riskier, but let's accept that there are different approaches in getting a molecule from A to B. Even once we get there, there are different ways of getting paid for an asset. And we saw during COVID, we continue to see with the GLP-1 market, there are many different ways to get paid. There are more ways every year as the US insurance market changes. And then maybe a very important one, but one that's often hidden, is this kind of range of risk. So we tend to talk about risk as if this was in kind of a single dimension, but clearly there's technical risk, and there's also hidden commercial risk. So launching a product that's not going to do so well when it gets there. But there's also regulatory risk, so getting something to the regulators which is turned down. All of those risks are apparent. So this huge range of stuff that we need to really tie down as soon as we can when we come to development. And this is where the kind of ingenuity of folks like the folks on the call today is really critical, because we tend to think about the molecule as if it was going to self-determine its own destination and get there by itself. But let's recognize there's a huge amount of work goes into even getting a molecule to be something that we can create in a lab, something that we can scale, something that we can build, something that we can put into the right formulation. And this Haber-Bosch story that we're all well familiar with, we get taught this at university early on, but it's often told as one of, as we said here, of insight by Haber. But the kind of the application by the businessman was probably more important. And as Matt Ridley again says, the ingenuity needed during Bosch's perspiration, getting this to work, getting this to scale, is something that we all recognize in our industry. That needs a lot more work than the simple discovery of a molecule does. So, I think that's important for us to understand and work on. So, come back to this asymmetry. So learning as a competitive advantage, understanding what might be approvable is often a task that our industry undertakes pretty well. Understanding what the product could be, so the dose, the device, the digital, the diagnostic, what the endpoints might be in our studies and so forth. Really, all of those things define the kind of classic product profile in our industry. But understanding what the market might want when we get there. And we're looking 10, 15 years out. So, we all are aware how hard it is to look so far out in our space. But, we're in an industry that's relatively easy to predict what's on the market in 10, 15 years time, because it's already in somebody's pipeline or it's already in the marketplace. And really our our target audience isn't going to change very much between now and then either. But so we say we still see opportunity in this kind of learning phase as a competitive advantage. And my kind of biggest passion is really to talk a lot about this kind of need to bring commercial and market access into early phase. And the pushback we usually get is, well, why would we invest that much in that work? Because the drug's more likely to die than not die. And I go, well, look, if we don't do that and it doesn't die, then what are we left with? We're left with a molecule that gets to market and doesn't really achieve anything. So it's this kind of classic paradigm of our industry, and mostly the biotechs are facing exactly this right now. So I mentioned the index, and I'll show you a little bit more about that on the next slide. But what we're seeing, clearly we're all aware of this downturn in biotech funding. Many great biotechs are trading below sometimes their own cash. That's probably not great for anyone. We need to really start to see the optimism that sits in there, because it's where pharma is getting most of its pipeline from now. So no one knows what's happening in the US. I know Vivek Ramaswamy personally, but I still don't know what's happening in the US and in the FDA. And we're seeing this kind of shift to de-risking assets and really clear commercialization pathways, rather than the kind of big challenging stuff that we're all more accustomed to if we come from the 90s or early 2000s. So this was our index this year, and this is the answer to the question: if you gave the same molecule to two companies before phase one, would they be equally successful? And you can see the innovation score along the bottom and the invention score along the top. But the companies that do well, AstraZeneca, Lilly, Roche, Merck, and some surprises there, like Pfizer, Regeneron, J&J have been there for a long time. Maybe as interesting as the stuff down at the bottom, so Merck, Takeda, Bayer, who haven't relaunched very much for a long time. But some companies that are better at commercial than they are at discovery, Novo Nordisk, I think you can all understand why they would have a positive commercial score, but a very poor invention score. And I'll talk a bit more about why we think that's important as we go through. So, this comes back to, is there an average pharma company? The answer is no. There's kind of really no such thing as an average pharma company. And that matters when we get into some of the kind of more interesting stuff as we go through. So I've highlighted J&J here because, year on year, we see changes. They were top of the invention score last year. Lilly, probably no surprise to anybody, have come top of the innovation index in 2025. And some companies that you probably expect to see in there, AZ of, Cambridge-based company, have done very well for many years now, top of the invention ranking this year, which no one expected. Remember 10 years ago when Pfizer were going to buy them and change them out of all recognition. No one expected AstraZeneca would become, bigger than it would even be possible for Pfizer to buy over time. We've seen the resurgence of companies like UCB. We've seen companies like Vertex, that specialist companies, so do very well. And interestingly, in the invention index, we've seen Beijing, so the first Chinese company ever to make the index, is starting to do well as well. And I'll come on to some of why this matters in the next couple of slides. I won't dwell on this because you can read the slide later if you want to. But people do well for different reasons. So Roche do well because they've had a lot of new approvals. Regeneron, quite the opposite. They've got the highest number of pipeline assets, but no new approvals. But they've had strong life cycle management with their biggest drug. People like Moderna, who did well during COVID, are continuing to do well. This reinvestment in revenue. And the comeback story of UCB, as I said, they've had these notable approvals, mostly getting out of epilepsy and a bunch of other places that they've been big forever. And then this kind of quiet giant of B1 or Beijing, who are very R&D intensive, really coming up, which not everyone expected. So the innovation index, invention index, this is just some of the breakdown of the metrics that we look at. So revenue from new products more than old products, the financial performance of the company, and where are they putting their money and how are they spending it. New drug approvals clearly are important, and key development events. So what's moving through from 2 to 3, and what's moving through from there into life cycle planning. Invention, all the things that you'd expect. So, things I mentioned, the novel regulatory designations, any key pipeline events, what kind of clinical trials are they doing, and what's the size of their novel drug portfolio. But this next slide kind of shows why, when we started looking at this more closely, why we started getting excited about the numbers that we were seeing. Because, what's represented here is really what we call the freshness index. So what proportion of a company's revenue comes from drugs approved in the last 3 years or the last 5 years, and the kind of red and the blue bars show you that. And this, there is no one company that you can pull from this and say, well, there's your average pharma company. Because there are, you'd expect some small biotechs to sit over to the left-hand side of this, because most of their revenue is going to come from drugs launched in the last 3 or 5 years. But you start to go down, you go, well, look, Lilly's there, Pfizer's there, Novo Nordisk's there, even Roche is trending towards the left-hand side. But some companies like Bayer, if you're Bayer and you're making nothing from drugs that you launched in the last 5 years, you start to think, well, we're going to have a challenge. There's 3 German companies sitting at the far end of this graph, where all of their revenue is coming from drugs that are older than 5 years old. And that becomes a challenge, because then we look at what we call fragility. So, which drugs are expiring in the years 24 to 28. And you can see this on the kind of gray and the blue bars here. So the gray bars are, what's expiring in the next 5 years. The blue bars are what we're making in 24 from drugs approved in the last 5 years. And then the kind of big metric is, well, what's the overall revenue of the company? So how big is the company in general? And you can see the kind of gap that some companies are going to have. J&J have a lot to replace, quite a high fragility bar, quite a small, 24 revenue bar. So, in the next few years, they're going to have really quite a lot to make up to maintain that kind of relative scale. So this is revealing, that there is no average company here. The companies that are the flavor of the month at the moment, Novo Nordisk, Lilly, they're in there. Lilly looks a lot better on this chart than Novo Nordisk does, simply because really a lot of their revenue is coming from new drugs. But it's quite a big percentage of their overall sort of company sort of spend. But then you come back to, well, what does that matter when it comes to R&D spend per approval? And you've probably seen this kind of expectation that is $2.8 billion per drug, because that's the number that companies like to use for the cost of development in their EMPV calculations. But look at this chart. No companies, well, nearly no company is spending $2.8 billion per approval. Most, BI, partly because they haven't launched anything, haven't had any approvals, are spending, a lot more than that. But among the companies that are launching drugs, $5 to $8 to $10 billion per approval is sort of reflecting that average cost. So, the average cost is around $7.5 billion per approval across the top 30 at the moment. But there are some companies clearly doing a lot better than that. UCB, I mentioned, have started to get things approved. Pfizer, relatively efficient and effective company, and Novartis is also sort of well reflected in here. So, I think this remains an important question for our industry is, why are some companies spending so much? And clearly not getting approvals is a bad way to do that. But, the companies in the middle are, AstraZeneca are, good performer on indices, but they're well around the average sort of cost of approval per asset. I won't dwell on this because this is a research paper where we put some of this longitudinal analysis of the data into the Journal of Pharmaceutical Innovation. You can all sort of go and take a look at that, but it says, higher R&D spend, lower R&D spend as a proportion of revenue, and the higher number of phase 3 clinical studies are the kind of things that tend to determine what you're going to make in the future, as you would expect. So again, I won't sort of dwell too much on that. Over the kind of years that we've been doing this, and we've been doing this for like 14 years now. In the last 7 years, we include 30 companies in the index per year, but that's, had companies come and go. So we've had a total of 40 companies in the index over the last 7 years. Pfizer and Lilly have done the best in the innovation index, 2 wins each. And AstraZeneca, as I mentioned earlier, are easily the most inventive company, because they've placed 5 times at the top of the index within the last 7 years. And the companies that you would expect to see have done well across multiple years. So Roche, Pfizer, Lilly, AZ, even BMS, which have certainly tailed off a lot in the last few years. So I thought I would finish up with this kind of return to that question. If you gave the same drug to 2 companies, would they be equally successful? Because it sounds like an abstract question, but it's clearly not. And I tend to use this case study to illustrate that.
Hi, Mike. Just to let you know, there's 4 minutes left.
Yeah, I think I'll be okay now. I'll continue to talk quickly. Keytruda is one of the biggest drugs our industry's ever seen. But you look at launch, 2017, they didn't win launch. Opdivo was ahead for a year and a half. Opdivo really was, the winner at launch. But Keytruda continued to build, life cycle extensions and novel indications. And, we've seen the same thing with the GLP-1 space. The Novo won launch, but Lilly have overtaken them with exactly the same kind of strategy. So, maybe winning too early is a bad thing. But both of those companies, Lilly and Novo in the GLP-1 case, have done much better than the competition. You look down at the bottom there, Tecentriq and Imfinzi have sort of caught up, but Bavencio from Pfizer and Merck, $3 billion cumulative revenue over the whole time that they've been on the market with essentially the same molecule. So, does it come down to anything other than decision-making in those companies? I'd argue that, you can't see this much difference in the molecules. You can only see this much difference by looking at the companies and the way they make decisions. So I won't dwell on this to kind of Noel's point. But I will say, look, this, many companies tend to forget what's obvious when they're marketing stuff, and they do forget the commercial is at least as important a part of the journey and the description of the product in terms of what you're going to launch as the molecule that you have in your hands. And look, AZ, because of where you're all sitting or where the meeting's based, it's performed consistently well, and you'd have to predict it's going to go on to even greater success with its high conversion of pipelines, especially in the worlds of oncology and CV. So that was about as fast a run through as I could give you into our insights into invention and innovation. If you want any more, you can definitely get in touch with me at this email address. There are white papers and a bunch of other things to download from our website too. So thank you for having me and allowing me to ramble. Are there any questions while we're waiting?
No, you will do the questions in the room, right?
This is one on chat. How would Mike evaluate platforms in biotechs? We're actually starting that exercise with about 100 biotechs at the moment. The challenge comes in that there's so many different exits for biotechs. You've got the biotechs like Regeneron and Alnylam that go on to launch themselves, often through collaboration. I think, Alnylam have only recently become financially profitable despite, many years of trading. And a lot of biotechs that are looking to exit and actually be acquired and sold to pharma companies. So we find it easy to look at invention in those biotechs, but it's much harder to look at kind of the creation of value from that same space. But we are looking at how to do it.
Thanks, Mike. I've got a question myself. if you're to talk to a young postdoc or PhD student who's finishing up and they're looking to join a pharmaceutical company, that would be, allow them to grow and innovate. Which of the indices should they look at on your graphs?
Well, I think if someone's looking for a career in R&D, I think the invention index is probably really interesting. Sort of set of data. Those companies are doing something meaningful in the space. They've got novel molecules. They're doing interesting things with studies. They're getting stuff to market. We focus on innovation just because really that pays for everyone's salary. But that tends to favor the kind of larger companies, the ones that are more commercially minded. But the world is an exciting place for biotechs at the moment, especially in the US and China. Is having a much harder time in Europe at the moment. So, but certainly, our white paper, I think, will give you a good clues as to, who's exciting in the invention space.
Okay, thanks very much, Mike. I would just like to say thanks again.
Thank you.
Okay, can everyone see my screen?
Stop. Yes, we can.
Yeah, good. Yeah, hello everyone. My name is David. I'm with Datagrok, a senior scientific developer, and thanks for having me, first of all. And today I'm going to be presenting the Cheminformatics part of our platform, Datagrok, which is a Swiss Army knife for all things life science, especially Cheminformatics. So my slides are going to be very small. Actually, most of the time I'm going to spend for the demos and actually showing our platform, which is browser based, and I should stress that it's fully free and open for academia and personal use. So anyone from academia or just for personal use just can go to our public website and enjoy everything that I'm going to show and much more after this presentation. So yeah, as said, my slides are going to be short, and then I should move to the demo. And before I start, quick intro to our platform. So as I said, Datagrok is a browser-based platform. It's mostly focused on life sciences, but it's very good at general data science as well. So you can access your data from wherever you want just by drag and dropping, connecting to databases or connecting to any file shares, pretty much anything. We read any file formats from basic ones, CSVs, then to more scientific ones, SDF and PDBs and so on. And then we have the whole myriad of scientific computations, more so in cheminformatics and bioinformatics, but also in other areas which I'm not going to go into today, such as differential equations and much more. So the limitations are very, very high. So, well, low technically. So you can have gigantic data sets opened up in browsers. Everything will be very interactive. You have the whole gallery of 50 or more different viewers, starting from the basic ones, scatter plots, bar charts, and so on, and then more scientifically aware ones for molecules and more proprietary ones. And the way it is built is that it has a proprietary core, which basically runs the platform, and the idea behind it is that everything is packaged as a function. So everything that does the computations, does the analysis, does the visualizations, and so on, everything is sort of a building block, and these building blocks can be added by anyone at any time. And the platform has a plugin system. We have over 50 different open source plugins, and Cheminformatics support is also one of the plugins, and then some of the other ones also extended a little bit. Those are all open source. We have some contributions from outside as well, and everyone is also always welcome to contribute. We have quite high adoption in the industry, especially in big pharma, as the biggest thing we do very well is bioinformatics and cheminformatics. And as the last speaker mentioned, J&J is one of our clients as well, and they also have very big adoption of Datagrok inside. They use it for various purposes, including cheminformatics, but in other areas as well. Again, as I mentioned, absolutely free for academia. We always encourage everyone from universities or any sort of academic background to use it. Completely free, and we also always like to support this usage. And yeah, as I mentioned, Cheminformatics is just another plugin that can be extended. It's day by day extending, adding new functionalities, new sorts of analysis, new visualizations, and so on. And as I said, it is running fully in the browser, and therefore the speed and performance is one of our biggest priorities. And what powers it, the cheminformatics part, is mostly RDKit and OpenChemLib, which most of you should be very well familiar with. It's basically using the latest technologies there are. WebAssemblies, the RDKit is compiled in WebAssembly, running in parallel web workers. So basically everything is super parallelized, and everything is sped up as much as possible. We use it for rendering, for calculations, for scaffolding, searches, and all sorts of different analysis. And then, as I mentioned, we have fully exposed APIs and fully documented workflows on how to develop things on Datagrok. And there are lots of community built functionality on top of that, apart from our own as well. So this has been it for the presentation. I'm going to now move on to the demo. Hopefully everything goes fine there. So I'm going to close the presentation and open the platform. And whenever you open it, basically it looks something like this, where you have multiple ways to the initial step would be then to bring your data. So as I said, you can just drag and drop the data from wherever you like. So for example, I can just drag and drop the CSV file here. And the first thing that happens is the CSV is read and the columns of the data tables are recognized. So recognition means that we detect if there are any special types in those columns. So for example, the first column here was detected that it contained mole blocks, or they could contain smiles or whatever. And instead of just showing smiles, they are rendering molecules. So this is very small data set of 1,000, but you can see the speed at which I hope the I hope the screen sharing is translating this correctly to you, but basically you can scroll through it very fast, and everything is very, very fast indeed. Then everything is very interactive and everything is live. This means that these are not just images. You can click on anything. So for example, I can click on the second molecule here, and on the right side, I get the context panel, which basically gathers throughout the platform all the computations, all the analysis, or all the properties that might be related to the molecule. So some of them are dynamically calculated, like this one, for example. This is a Python script that is written in Datagrok, and it returns an image of partial charges. Then we can have some properties calculators, which calculate some different properties. We can have descriptors. We also have the retrosynthesis path predictor. We have database lookups. So these are some of the default databases that it comes with, but again, anyone can plug in their own database and have a lookup of the molecule there, including substructure lookups, exact or similar lookups. And again, there we have a bunch of different properties, like biological ones, structure-related ones, and so on. The other thing that it does, it unlocks the Cheminformatics top menu, which has the whole suite of different analysis and different calculations that can be applied to the molecules. So this starts from docking, for example, different analysis, including hierarchical clustering, chemical space, which is UMAP or TSNE based on the molecules, elemental analysis, R group decomposition, activity cliffs, structural alerts, scaffold tree, matched molecular pairs, and so on. We also have the similarity search, diversity search, substructure search, which are also very, very fast, accelerated to maximum. I'm going to go into details on all of these in a bit. We can calculate a bunch of stuff, like, again, as I mentioned, descriptors, identifiers, clustering, MCS for the molecules, translate it to different notations like INCHI, calculate other properties, toxicity risks, MPO scores, biochemical properties, and we can transform these molecules in any way, shape, or form, like converting notations from smiles to mole blocks to different version of mole blocks to smarts. We understand the platform understands anything, and we can run different sort of chemical reactions on them as well. But first, I'm gonna start with some of the searching, and then I'm gonna go into the analysis part. So as I mentioned, you can bring quite a large data sets into browser and visualize it and analyze them without a problem. And well, as much as the browser and your computer itself permits. So most of the heavy lifting is done on the client computer itself. So here, what I have is a data set of 1 million molecules. And again, as you can see, no lag at all. I can just scroll through it and see any of the molecules. And one of the things I might want to do is, for example, a substructure search in 1 million compounds. I can do it very easily just by opening up the filters panel and clicking here on sketch for the molecules. And as soon as I sketch something, you can see that the structures containing the given substructure are filtered out. The given structure is also highlighted. And again, I can just scroll through it and visualize everything without any noticeable lag. So whenever I just redo this, immediately everything is recalculated. So you can understand what sorts of speeds we are targeting here. Right. So now I can move on to the different analysis. And for this, I have prepared some of the different demos here. So for example, one thing that is very useful in analyzing large chemical spaces is the chemical space itself, or the visualization of it. So something like a UMAP based on fingerprints, which is something like this, where it can do the clustering, it can do the embedding, and then the clustering on top of that, and then basically seeing what similar molecules end up together. And in the same area, we also can do hierarchical clustering, or visualizing it with a tree. So I can just narrow it down like this. And similarly, you can see that everything is very, very fast, responsive, and so on. And to prove my point that this is actually done live and on the client side, here I have another data set of 100,000 molecules. And what I'm going to do is try to run a chemical space live, something that probably that takes a quite a long time. So here, basically, okay, let me adjust this parameter that I know is better. So I'm going to click on the run, and it's going to complete in under 30 seconds. And in the meanwhile, I'm going to explore what this chemical space offers. So you have an option to configure what fingerprints are used for the similarity calculations, and then what distance function or similarity function is applied to those fingerprints, what method you want to use, is it a UMAP or TSNE, and adjust all sorts of different UMAP parameters. And also, after the UMAP is complete, you also can do clustering based on the embeddings. And then based on those clusters, you can also do the cluster MCS, which basically goes through the clusters and calculates the most common substructure of those clusters and puts them in the molecules. So here we have pretty much it completed in, as I said, under 30 seconds. And let me adjust the size of the dots here. So here we have very large chemical space, which is clustered in some way. and it's put in a scatter plot, which is, again, as everything else at Datagrok, very, very interactive. So I can zoom in, drag and drop at any place. So for example, I can zoom in somewhere here at a particular cluster, and I can see that in a cluster, the molecules indeed are very, very similar. So similarly, I can zoom into any other cluster, can just disable labels here. And for example, in here, and on the left, I can see that all the molecules in the cluster are indeed very, very same. So this, again, is something very useful for analyzing large chemical spaces, where a good representation of embeddings in a 2D space is very visual, very informative, and also something that requires tweaking the parameters, something like UMAP parameters or the distance parameters, and you don't have to wait for hours and hours for it to complete. So in Datagrok, you just do it in under a minute. Then going on to different analysis, we have similarly the activity cliffs analysis. So this does the same clustering, but also takes into account some sort of activity column. So this can be inhibition towards something or pretty much anything else. And it calculates the, so it basically draws the lines between the points, which are structurally similar, or rather their fingerprints are similar, and but their activities differ quite a bit. And you can see those lines here. So I can zoom in somewhere here. I can just hover over the line. It's going to show me the difference. Just click on the line, and the same difference is going to be shown on the context panel here. Then we have obviously the R group decomposition analysis, where we can just draw out a core of, if I have some data set, I can just draw out a core, and based on that core, then I can perform the R group decomposition and generate all sorts of different visualizations, like a trellising of this based on R1, R2, or I can just with a single click change this to any of the other columns and visualize it via a histogram, or maybe a bar chart, or maybe some other line chart, and also generate other sorts of visualizations. And the UI for this is also very, very easy. So you get a very easy dialog where you can draw out the core structure, or you can just click on the MCS button, and it's going to go through all the molecules and calculate this most common substructure. Next up, we have the similarity and diversity search. So this basically goes through the whole data set of molecules, calculates the most diverse structures, which is down below, and then based on the molecule that you click on, it's gonna generate 15 most similar molecules to that. So for example, on the bottom here, I have 15 most diverse structures in this given data set. I can click on this one, and you can see that the top one updated based on the similarity to the original molecule, like so. And same can be done from the data itself. And on the bottom, you can see the very similar chemical space of this data as well. And then for searching, something that is very, very useful as well is scaffold tree. So you can draw out different scaffolds that can be useful in your data set. And then based on these scaffolds, you can color them, or color the molecules, or color the fragments within them, and also filter them. So I can just click on these, on this, and something like that. And I'm going to get the molecules that correspond to the scaffolds or molecules that contain these scaffolds within them. Now, what I want to spend a bit more time on is matched molecular pairs analysis, which is more comprehensive type of SAR analysis for molecules. And this is, again, fully running in the browser. And in this case, what I have is a 20,000 molecules, where what happened is it went through all 20,000 molecules, it broke them down in different ways into fragments, and then starting matching the fragments and cores between different molecules, generated the transformation rules, and then based on those rules, we have different sorts of visualizations and analysis results. So for example, here, what we have is the substitutions tab, where I can see the from fragment to fragment transformations. If I click on them, so for example, if I do like this, if I click on them, I can see the exact molecules that underwent this transformation, what was the activity change associated with that transformation, and well, again, there can be multiple activities, as you can see here. So we have some different properties that underwent a change under this transformation. So here, for example, if I sort by total count of transformations, I can see that there is a lot of pairs that have methyl to benzyl transformation. And on the bottom here, I can see all of the exact molecules. If I click on them, I get the visualizations on top pinned. And I can search through any sort of transformation and also associate the average direction of activity or property that is associated with this transformation. Then I have the fragments tab here, where I can see the trellised version of the similar information, and it shows in a bar chart what is the average property or activity change that was associated with transforming from this fragment to this fragment. And this, I can just take this and scroll to anywhere, and as everything else, it's very interactive, and some good conclusions can be then drawn from here. And this can be filtered as well, and so on. Then we have the Cliffs tab. Then we have the Cliffs tab, which is the same as the activity cliffs that I showed you previously, but it also, but instead of relying on the molecule similarity based on the fingerprints, it relies on the same core but different fragment transformation. So every arrow is corresponding to a concrete transformation. So for example, if I zoom in somewhere here, this arrow is corresponding to a oops. Yeah, I cannot click. Okay, so this arrow is corresponding to a certain transformation, and not just the fact that these two molecules are similar. And then basically, since we have generated all the rules for the substitutions, we counted all the fragments, we know all the averages, and so on, we can draw some conclusions from it and do a quite mechanistic and statistical generation of new molecules, which is done in the generations tab here. And basically what it's doing is, for a certain core, we try to analyze what sort of transformation would potentially lead to the maximization or minimization of a certain property. And here, as you can see, we have generated about 40,000 different new molecules. And here is the predicted property value that is associated with this prediction. On the right side, we can also see the scatter plots that are the correlation of observed versus predicted, and therefore can we use this model of transformations to apply to different molecules as well. Right. So we can go into the MMP if someone has any questions a bit later. But generally, everything that I was showing, as I said, is inside the browser. This everything is very extendable and is modified on a day-to-day basis. And modified, I mean mostly improved. So we quite work a lot directly with the big pharma, and this gives us a very good understanding of actual use cases in the pharma and how these stuff might be used. So basically, we are firsthand getting the information about what is useful for different sorts of analysis, and this is exactly what fuels the development on our side. And this MMP was one of the good examples for that. We are working on different other different sorts of analysis. Might be very useful to hear about the suggestions, what and how we can extend this. But yeah, and I guess on that part I can conclude, and we can move on to the questions. Mostly I wanted to focus on cheminformatics, and there are much, as I said, there are much more things in other domains like bioinformatics and mathematics and so on, but I'm not going to go into that. So again, thank you very much. So guess I can finish on that.
Thanks very much, Tavs. So, any questions from the room first? Yeah.
Thanks very much, Peter Murray Rust. That was a very impressive system, both in terms of its functionality and its speed. I think at the beginning you said that the plugins were open source. Now, I could have misheard that, and it could be that they were built with open source components. Could you explain, whereas your core architecture is closed? So could you explain how the plugins works and what the access to the various open source components are and whether they're standalone?
Yep. So both are true. Plugins use open source software like RDKit, OpenChemLib, and so on, like the Cheminformatics plugins, and the plugins themselves are open source. So whenever you go to our, let me share again then. Whenever you go to our public version of Datagrok, which is at public.datagrok.ai, which is here, every plugin that gets pre-installed, those are open source and they are under our GitHub. We will share the link to this after the presentation if you would be interested. And basically how it works, what I meant by proprietary core is that the core is the system that connects these plugins together. And what plugins do is they expose a certain functionality for the platform. So the plugins tell that I have a function or
David,
I have
you're
a
showing
method.
the wrong tab, I think.
Oh, what am I sharing?
You were sharing the thank you screen.
Oh, okay. Oops. Okay. So, yeah, sorry. So everything is under our Datagrok AI public. But if I would to show the plugins, so here is the list of all basically open source plugins which are under the same GitHub. And what these plugins do, what I was saying, is that they tell the platform that I have this function can do this function has a certain role. So for example, one example that I showed is that when you click on a molecule, it's very context aware. So this right panel we call a context panel, and a function inside of the plugin can say that I am a context function and I'm applicable to molecules. And this is all the platform needs to put the results of the function in this context panel. And this is exactly how these properties work. And this is just one function inside of the Cheminformatics plugin, which just says that I am a context function, I'm applicable to molecules, and I calculate some properties. So put me in there. This one here is actually just a Python function that is running on. So from what I showed, this Gastiger partial charges is the only thing that is running on the server. Everything else was in the browser. But this one is just a Python function. I can even click on it and see its content. So what it's doing is taking the molecule, drawing the graphic of Gastiger partial charges, and just returning the graphic to Datagrok, and then it gets put there. I hope I answered your question. Didn't move to the tangent line there.
I'll add just a little bit. Yeah, hi, I'm Andrew. I was supposed to do the talk, but I got some bug. So you can think of Datagrok as an operating system like Windows, right? It's proprietary. It could be free for someone, but there are tons of open source applications for Windows, right? So yeah, the same concept applies here.
Yeah. Any other questions?
So thank you for the talk. So I just want to ask, from a perspective of a young researcher, like how do you, like the conception of Datagrok, how do you identify within the area of like cheminformatics and software? How do you identify like the niche, and how do you concept the idea of Datagrok?
I would say basically the platform for life sciences, meaning that you can, we are moving and always trying to be the platform so that you wouldn't have to move anywhere else. So you bring your data, you read your data, you understand your data, you calculate new data, analyze your data, and we also have machine learning models, and we can train and run machine learning models inside of this platform. So basically, the idea is that the platform where you can do everything that you want in terms of analysis for cheminformatics, bioinformatics, and whatnot. Andrew, I don't know if if you
Yeah,
if you
I
do
might take the second question regarding the conception. Yeah, since that was me many years ago. So before founding Datagrok, I was with J&J for 17 years. So most of my career was building analytical systems for cheminformaticians, bioinformaticians, later clinicians. So eventually it was just very clear that in order to truly make something great, we need to first start a new company, and second, build from the clean slate without the historical graph. So yeah, we did just that, and here we are, almost 7 years later. Yeah, I think we achieved the objective. So now we want to expose our platform to everyone. In particular, we think academia would find it very useful. We certainly hope so.
Okay, yeah, thanks very much. Any other questions here? Okay.
Hello. Am I correct that you need to upload your data to your cloud? It's not your data doesn't stay only locally and is processed locally. It's going to someone's cloud.
So it depends. So first of all, academia can use either our cloud instance, or academia can also just download the Docker containers and run the platform locally. So we offer that. But even if you run it on the central instance, in many cases, you don't need to upload the data at all. Like what David was showing in his example, I don't think we touched the server at all. The data stayed local to the browser. We make it super easy though to, sorry, to upload it and share with people. So that's super easy. But by default, we do tons of things absolutely in the browser. So the data that David uploaded from his local drive, it stayed on his machine during the demo.
Okay, thank you.
Yes.
Yeah, thanks very much. Okay, one last.
Hi. Hi, thank you for the presentation. Nicola Boss from EBI. I know that you're using Sure Campbell data, and I was wondering if, for patent information, I was wondering if you can tell us or maybe briefly show us what you do with this data, please.
Yeah, precisely that. Actually, it's like it's in Campbell. In Sure Campbell, it's you can do substructure or similarity search, and it's going to look up the molecules and the patterns associated with the molecules that have a similarity to this one, or a substructure for a given one. So for example, I just clicked this. I searched by similarity. There are 4 patterns found. can just click on the plus, and it's going to open up a new data that contains the molecules, the patterns with links, and so on. And again, this is just another plugin, and inside of the plugin, it just tells that I'm a context, I'm a context function, and I can look up the patterns for molecules.
Yep, very nice. Okay, then, thanks very much for your presentation.
Yeah.
Just thank the speakers again.
Thank you.
So, yeah, those of you who are online, if you want to stay on, and Andreas will organize what's the word for the Zoom thing? A Zoom room, Zoom breakout rooms, if you want to have a chat. And those of you who are here, there's been a late breaking announcement that the Panton Arms no longer has food, but the Alma does. So please join us at the Alma afterwards.